Yes, good evening to everyone. Welcome to the day 9 session on molecular talking analysis and MD simulation. Before proceeding to today's session on day 9 session on molecular talking analysis and MD simulation, can we recap the last session topics that's on molecular talking? Now, let me take you to the polling. Okay, so through polling, we can have an interaction. So we can start the first poll. So I just started the first poll, which is not a talking software. The choices provided are Glide, Flex, X, Chromax, and PYRX. So try to answer for this question, which is not a molecular talking software. Only one is not a molecular talking software. Actually, like GAT examination, which is a common entrance examination to get entry into IITs and NITs. Similarly, you can see we have examination like BINC, Bioinformatics National Certification Exam. In that, we have purely bioinformatics subjects. Whatever you study in computer drug design, this is of course coming as a core. Got it? Like if you clear BINC examination, Bioinformatics National Certification Examination, you can able to get entry into IITs and NITs. This is specifically related to the bioinformatics. So of course this is most important, and so these type of questions we will be anticipating. Okay, and I think even the GAT 2027 is going to be conducted in IIT Madras. I believe you have received this information. We have GATE Biotechnology, GATE Excel, Life Sciences. We also have thermodynamics separately. Thermodynamics is a separate category like biotechnology. So we also have bioinformatics through Bioinformatics National Certification. Very good. So I found only one answer from your side. Okay, so out of the time provided, everyone answered Chromax. You are right. Then what is Chromax actually? Chromax is MD simulation software, isn't it? So how about the Glide? Glide is from Scroidinger. Okay, and we have FlexX and Pyrex. I think you know MGL tools, Autodoc. PYRX. Of course, that is a virtual screening software, but that is a talking software. Now, I'm going to launch another poll. So, see the question, which is not the model or theory of molecular talking? Which is not, it's not a mode, it's a model. Which is not the model or theory of molecular talking? The choices provided are conformational ensemble, Monte Carlo, induced fit, lock and key. Good interaction, but how about others? One day I'm getting 8 to 9 words. Yes, 10 words, very good. So just last 5 seconds remaining. Try to answer anything. I can't even see your names. There is nothing I can able to see. One day I'm going to see the answers around. So now I'm going to freeze this. I'm ending the poll. The answer is Monte Carlo simulation. Your answer is perfect. Okay. It is coming under simulation model. It's not a talking model. So for molecular talking, we use Fisher lock and key, post-land induced fit, and conformational ensembles. Okay, so these are the methods we use where it uses the theory of molecular talking. Okay, I think many of them understood molecular talking properly, so only you are providing very correct answers in all the poll. Now, let me, before starting today's session, can we have one participant question like yesterday she raised that she has having problems in working with Discovery Studio Visualizer, I believe. So, are you available? madam.
Yes, sir. Yes, sir.
You better share your screen. I just coming out. I stop sharing. Let me see how I can able to clear your doubt.
Okay, sir. I'm sharing my screen, sir.
And similarly, participants, if you have any other doubts, okay, let me know in advance. Okay, at least one day in advance, so that we can have a sessions. I mean, like this. Okay, to share your screen and to discuss. So after this, I'll be taking you to how to analyze the results of molecular talking through Autodoc. You remember yesterday I worked with molecular talking through Autodoc, and I've completed up to 6 steps. The seventh step is to analyze through Python PyMod. You know the tutorial of Autodoc is available in the folder I have shared to you. Not only you can find the demo files as well as installation files, but also the files to be worked. I told you in a desktop you have to create a folder, can be a name like a demo, and inside the demo folder you have to paste the Autodoc tutorial, Autodoc 4.exe, AutoGrid 4.exe. Then you can paste the ligand and receptor 3D structures. Okay, so these are the 5 files. Initially we make it in a folder, and then we start doing talking by fixing the location. Initially we have to fix the location, then only we can able to see all the intermediate files are going to store in that location. Okay, so after that I'll be taking you to the MD simulation. Discovery Studio Visualizer, if you see in my chat box, okay, the latest thing, Discovery Studio Visualizer 2025 is available. Whatever I have kept in my folder, that is going to take very less storage. For that, that is actually an Astralis product. But now at present, it is a BioWare product. Okay, you're getting something like program maintenance, repair, remove, modify. Can you go to back? No, 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 not this back. Yeah, that back. I think you have already installed. Back, again back. Welcome to the maintenance installation. The installation will slow you to modify, repair, or remove your existing. So I think you can click cancel. I don't know. We can check how yes or no, or you want to cancel. The wizard was interrupted. Finish. Now, can you go to the Windows and type uh, Discovery Studio? There in search. Discovery Studio client is there, isn't it? Click the Discovery Studio client. So after installation, click no, no. So see, the Discovery Studio Visualizer is going to get open. Actually, the Discovery Studio Visualizer is successfully installed in your system. Only thing is that we don't have a license. So the visualizer can be accessed without the license. So if you're opening the Discovery Studio, then you have to click no. Like even in Autodoc, for the first 15 days, you don't ask anything. Cannot create directory, invalid document. Click OK. Yeah, see, the Discovery Studio Visualizer is open to you. The license we have to pay, okay, around 20 lakhs. So, but whatever I practiced, okay, and whatever I'm going to practice it on today, like how to analyze the results, this I can able to work with this Discovery Studio Visualizer. Okay, now I think your problem is solved.
Yes, sir. Thank you so much, sir.
1 minute. Stay back. Just you can able to you have to do one more thing here. Can we go with the one animation? There is one animation called build a protein structure. Can you click that build a protein structure? The third one from Quick Tours. Uh, See, it's directing you to click yes now. So, like if you want to build a protein structure, it is asking. Now, again, you are getting one more prompt. Okay, to start this, you have to click yes. That's what they have mentioned. So, default, you click yes once again. So now it automatically it does. Okay, so no need to select everything. This is something like an animation already developed. Okay, and you have to click yes. So I think it's automatically done now. So after clicking yes, even getting a prompt box and what to do, automatically the cursor go and go to the clicking. Now are you going to click or it's automatically moving?
Sir, I clicked only yes, sir. Remaining
Yes, so
automatically coming.
automatically it happened. You got it. See, she is not doing anything. I am not doing anything. Whatever the animation they have done in Perl scripts, this is automated. To build a protein structure, it is doing all these steps actually. You got it? So in Discovery Studio Client 2.5 from Asterisk version, you can able to see these kind of animations. So not only you can able to build up structures of protein, chemical compounds, and analyze the results of analyze the molecular docking results, but also you can animate it. If you want to provide a demo, you can use the Perl scripts and animate exactly like how it is done now. Okay. So okay, good.
Sir,
You
what
also
about this thing, sir? An error was encountered.
actually, you don't have to worry for this. Okay, nothing is going to affect your system. Simply have to click OK. And now, can I go ahead by sharing my screen? If you stop
Yes, sir.
sharing,
Yes, sir. Yes, sir.
ah,
I'm stopping.
ah, kindly move. So now I'm going to share my screen. Okay, so shared my screen. Now let me show you from what exactly we have terminated on yesterday. Actually, I was explaining you like to create a folder called demo 10. Then I have had just 5 files. You remember? I had only 5 files. Okay, like Autodoc 4.exe, Autocreate. Even this is available in the folder I have shared to you. Just like that, copy and download, and you can use this, store it in this folder. If you do it like this, it will be easier to complete. See, we have generated so many intermediate files doc1.log doc log file, and this is doc parameter file, grid log file, grid parameter file. So these files are generated intermediate when we followed the 6 steps. Okay, also map files are generated. And if you want to see the final file, how we have saved this file, internavir underscore protease underscore complex dot pdb. You remember? This is the final file I have saved. And participants, my suggestion is better work on the same day or on the immediate day. If you if you're not practicing on the same day or the immediate day, the next day, you will be lagging behind. You don't know where exactly you caught in if you are not getting a result. So try to complete your task. Okay, whatever homeworks I gave. Only 1 hour I trained, but I believe I've given a lot of inputs. Okay, then only thing is that you have to try the remaining 23 hours or from the time of sharing. Now let me take you to the complex file, whatever we have created. I told you we have saved like PDB file. Let us check whether it's in PDB file extension. Yes, now you can see this. This is in PDB file. Okay, so we have to simply double click. I have installed Discovery Studio 2023, and the link I have shared you in the WhatsApp as well as in the chat box is 2025 version. Even that is a freeware, so it looks like this kind of look and feel. Okay, and see whatever that Internavir ligand. So this is the Internavir ligand. Okay, which is talking with the protease. What protease is that? HIV protease. Now go to the display style, graphics, right click here, going to the graphics, clicking white to get the background as white color. Now you can see for the publication generally we need these kind of very clear outputs, isn't it? So now we can see this, and but if you want to see further the interactions, see the first step. What I'm going to do here is go to the receptor ligand interactions. See at the top, receptor ligand interactions. Here you have to simply click show 2D diagram. If you click this, you will be getting this 2D diagram. So in this 2D diagram, it is evident that we can able to see the interacting amino acids. The interacting amino acids of the internavir or the proline 1 cysteine 95. Then what is A here? A is nothing but the A chain. We have only one chain in the protease, and we just tried to talk with the engineer, and we got this kind of a result. Whatever best conformer we got, the best conformer what we got has the score of around minus 6.28 kilocalorie per mole. This is the energy we got on yesterday. I told you if we get energy minus 6 and below, then it is a significant thing. Only the conformer 1 we are getting the significant. If you see the conformer 2, it is minus 4.12, which is not significant. So, but whatever default I have taken, the first conformer, I saved that ligand underscore receptor PDB file, and I'm just going to do the analysis based on that. And I told you this is Ki inhibitory constant. Okay, so the concentration value is mentioned as 24.84 micromole. Of course, these two are important data here. Okay, now let me take you to this Discovery Studio. I was explaining you that this is a 2D interaction diagram. Earlier, whatever we see is seen in the black background converted to white background is a 3D interaction diagram. Okay, and you can see not only the hydrogen bond, that is conventional hydrogen bonds, we can also see few more bonds. Exactly, if you see, we don't have conventional hydrogen bonds, but we have that green color bonds, whatever that aspargine 98 is a van der Waals bond. Okay, we have a van der Waals bond. If it is a light green color, it is a carbon hydrogen bond. Carbon hydrogen. This is not a van der Waals. This is looks like a carbon hydrogen bond. Okay, and similarly can find a pi sulfur bond in orange color, pi bond. So many bonds you can able to see. From the legend you can able to find. Okay, what are the interacting amino acids and what are the types of bonds? Now, if you want to see this in elegant manner, I simply right click here. Okay, go to the atom. Now, presently we have the ligand in a line model. But if you want to have in the form of a stick model, I simply click stick. Now you can see the stick in that is ligand in stick model, and residue if you want to be in a disk model, click apply disk. So earlier we had a ball, now you can see it's in very contrast disk is formed on the surface. Now the most important one more thing I have to show is the interaction. In the interaction, we don't find any classical interactions. The classical interaction is nothing but the hydrogen bond interactions. Okay, we don't have the conventional hydrogen bond interaction for this interaction, intermediate binding with the protease. But we can able to see the non-classical interactions. The non-classical interactions are this carbon hydrogen bond and other kind of a bonds. Okay, in hydrophobic bonds, you can see pi hydrophobic, alkyl hydrophobic, all these things. So these are the bonds actually present. But here, why I'm showing you in the interaction, if you want to measure the distance of the bonds, click show distance. If you check this show distance and click apply, you can see even the distance bond lengths. Now see the diagram how it looks like. Earlier the 2D interaction is not looking very clear, but now it's clear, isn't it? The ligand in stick model and the interacting amino acids in disk model, and even the hydrogen, the bonded interaction lengths are also shown here along with the legend. So this is how we can take a snapshot and we can paste it in the publication after the docking. Or else even we can able to show this 3D interaction. From this 3D interaction, if you want to show the results, okay, how to show the results? For this, we have only 3 steps to follow. The first step is go to scripts and then selection, select ligands. If you select the ligands, this is now selected in yellow color. Earlier it was in cyan color. Okay. So, and one more thing, you have to click view hierarchy. If you click view hierarchy, you can able to see the description. Okay. Like whatever you see the A chain is the A chain of the HIV protease. We can able to see around 99 amino acids are present. This is a 99 amino acid protein and only one chain is present. So only we can able to complete the docking as soon as possible on yesterday. Not only the receptor is a small molecule, even the ligand, intinavir is also a small molecule. So the docking speed depends on the size of both the ligand and receptor. So whatever you can see, this MK1902 is nothing but intinavir. So this engineer is present in chain, but whereas the protein is present in chain A. Okay. So I just mentioned you like if you want to see any kind of interactions in 3D, so first you have to go to the script selection, select ligands, isn't it? That's what we have done. You got yellow color. Now we have to select atoms within 4 angstrom. The second step is to go to select atoms within 4 angstrom. The objective of why we go to the 4 angstrom, if you want to check the binding sites near to the 4 angstrom area, then we can go for we can select this atoms within 4 angstrom. So now I'm going to select this. So after this, we can go to the scripts, ligand interactions, show ligand binding site atoms. This is the third step I'm going to make. View hierarchy you can open initially, then you can do these three steps. If I do these three steps, you can see hydrogen bond monitor. In hydrogen bond monitor, the ligand is MK1902 as I told. It is interacting with isoleucine 3, okay, of a chain. Isoleucine of third position of a chain is interacting with the ligand. Then if you see the interacting atom, this is very small, I can't able to zoom in. But I can able to take a snapshot and I can show you, okay, in some image. Power PNT. Do I get a blank PPT? I think it's taking some time. Or else I must paint. Now I just zoom in. I'm pasting this. So see here, whatever you can see here, the ligand, okay, of N5 atom is interacting with the HN of isoleucine 3. Plot it, whatever you find initially as the A is the A chain, IL3, HN is interacting with it. So this is of course one of the data. After the talking, you can able to take 2D interaction image as a data, 3D interaction image as a data, and whatever data we got for a binding energy Ki, and these interacting amino acids and the atoms of interacting amino acids and the interacting uh, ligands, these are also of course considered as a data. Okay. And but the 3D interaction, you don't find much thing, but you can able to see only isoleucine alone is the interacting amino acid. So what I'm going to do here is I am going to select everything. So yeah, our entire protein molecule is visible now. But isoleucine alone I'm be selecting now. Okay, and then what I'm going to do here is I click structure labels add. In the structure labels add, I go to the amino acid in object. And in attribute, I go with the amino acid and insertion ID and insertion code. 3D letter ID and insertion code. And I going to increase the font for the label, whatever I'm going to give. The label of the font size I provided is 24. Earlier it was 10. Okay, now I simply click apply, click OK. So if you see here, isoleucine 3 is visible. But isoleucine is in green color. If you want to change the color, even the color, you can able to, again you have to click add instead of green. You want to get this kind of a color. Now just apply. So now if you see this, the label isoleucine 3 is in this color. So if you have multiple interacting amino acids, you can able to find that. You can able to select that and you can change it. Now, whatever isoleucine I have selected here, if I want to display that amino acid, so I right click in the display style, and here I'm going to give like ball and stick. You see how it looks like now. Note this. I want only this 3D. So I don't want even the hierarchy presently. If you want to close the hierarchy, close hierarchy. So see this interaction, how it looks like. The entire protein molecule, okay, uh, is interacting. The isoleucine 3 is the isoleucine 3 is interacting with the ligand, and this green color is the hydrogen bond, carbon hydrogen bond. Can you see this? If you want to increase the font, so this is how you can take a snapshot for your publication. Okay, ligand in one shape, and the amino acid protein is in another shape, and this font also can increase. Okay, so these are the types of data one can able to see here. Now only the PowerPoint is getting open. Okay, now let me take you to the docking analysis I have completed. Now let me take you to the PPT. Like today, we'll be discussing on Discovery Studio analysis. I mean, talking analysis is in Discovery Studio, just now completed. We can see some publications and how we are taking the data, how we are inputting the data from the analysis. And then after that, we go with what is MD simulation, how we can able to perform with a free pair called Gromax. Gromax is a free MD simulation software. Okay, but we have some disadvantages to work with MD simulation. We can't able to work MD simulation in our laptops. We can't even work with desktops. We can able to work only with the workstations. Workstation, I mean, you have to use larger capacity for storage as well as more processing power. If and if you have a high processing power, like it should be like a server, so at least 5 to 10 times capacity. Okay, the storage capacity should be 5 to 10 times when compared to the normal CPU, whatever we use, and even processing power should be up to that. Then only we can able to work with this Chromax. And of course, we should have a high configuration graphics card also. We should go with 6 GB or more than 30 uh, GB graphics card to work efficiently with the MD simulation. We also have commercial tools like Discovery Studio simulation, Crowd Engine simulation to packages. Okay, but actually that is we have to pay annuals. For that, you can able to work in Windows itself. But for working with Chromax, you can work only in Linux operating systems, and that too in workstations. Okay, if you want to work with Windows, you have to install a interface. Then only through the interface we can work the AMD simulation using Chromax. Okay, now let me explain you further more about that. First, we discuss about the molecular docking analysis, like a few citations of literatures. The first literature I'm going to show you is this. As I believe we also discussed about this kind of a literature, that is docking a plant compound with A, which is a steroid electron of ashwagandha. I just talked with 8 different receptors, and out of 8 receptors, I found the negative binding energy only for these 2 receptors when compared with control drugs. For PARP1, we have one control drug, and for P2Y1, we have another control drug. When I compared with the vithaferin binding energy with these two control drugs, I can able to see the vithaferin A is showing negative maximum binding energy. So only I'm choosing these as the best targets for the vithaferin A. These targets are important for the cardiovascular diseases. So for in this journal paper, I tried working with Autodoc for docking and Chromax for simulation. Now let me take you to the first thing, interaction energies of vitafine with targets. You see this, as I mentioned, I got only the PRP1 and one more thing. What is that? P2Y1. These two alone I have screened as a best targets. Why? No. So if you take this drugs, these are the drugs actually. Vitafrin A is compared with this 9 drugs. Actually, I didn't take 8 receptors, but 9 receptors. And so you got the 9 drugs. Out of these 9 drugs, when I compare, so the Vitafrin A has shown minus 10.4, which is negative maximum when compared to Rocaparib. That is a kind of a control drug, which has given around minus 10.1 kilocalorie per mole. Similarly, if you see to P2Y1, the vitafrin is given minus 9.2, but here the yield is less, minus 8.8 for MRS2500. The rest you can see the control drugs are giving a negative maximum binding energy. So I have taken this as the 2 important targets and did MD simulation with the docked complex. Like whatever I docked vitafrin A with PRP1, okay, the final complex file, however I saved like Internavir underscore protease complex dot pdb. Similarly, this vitafrin A PRP1 underscore complex dot pdb is saved. After that, that file I have given as an input for MD simulation. You got it? Output of docking is an input of simulation. Similarly, tried with P2Y1. And however, the interaction energies we got, I'm just tabulating in the form of bar charts. Also, we have one more kind of a score called DSX score. Okay, similar to the binding interaction energy. So this also is tabulated and given in the graph. And what are the important results we can show after the docking? As I mentioned, if I go with a view hierarchy and I have taken the three steps, we can able to see the interacting amino acids and the interacting atoms, isn't it? So vitafrin A, according to this, is the C8 atom of vitafrin A. There are plenty of atoms in vitafrin A, isn't it? This particular atom is interacting with the C5. Okay, I believe not C5. It is interacting with the O of lysine 370. Similarly, C33 of vitafrin A is interacting with the N atom of histidine 51 position. So like this, you can see plenty of interactions. These are the interacting amino acids with the vitafrin A for this receptor. And you can able to see the length of bonds are also tabulated. So these are the different scores one can able to find, one can able to tabulate. And I shown you the 2D interaction diagram for this, I believe, on yesterday. And this is another kind of an case study where I tried talking of polyvinyl, not just a single compound. I believe I tried with polyvinyl A to E. So totally five compounds. A, B, C, like polyvinyl A, polyvinyl B, polyvinyl C, like this up to E. Five compounds up to E. When I tried talking compounds, five compounds with totally five receptors. The five receptors I've used is vascular endothelial growth factor and endothelial growth factor, BCL2, caspase 3, and caspase 9. So if you see these five receptors, okay, so few receptors are responsible for antineoplastic activities. Some are responsible for apoptosis. Okay, so we have different markers for this hepatocellular carcinoma. And when I try docking these five compounds with these five receptors, okay, I can able to see the best compound which is giving negative maximum energy. The screened one is the pyrimidine B. And the when I compared with the control drug what I used is the sorafenib. This is the best drug used to treat up to the second and third stage of cancers, of second and third stage of liver cancer, sorafenib. This is a multi-kinase inhibitor. And whatever we have used this sorafenib and we compared with this Peruvian B, so the Peruvian B has given significant results. I mean, comparatively better results with caspase 3 and AGFR. So only I've chosen these target receptors and I've done further things. So see here, the vascular endothelial growth factor receptor. When I docked with a PV, I got negative maximum minus 8.04. Similarly, tried with caspase. The remaining thing we are getting significant energy, but not much difference. Here you see minus 8.04, minus 6.45. So you have more than around 1.5, isn't it? Because we have vast difference in energies, I just scrutinize this as the best important target receptors. And these were the interacting amino acids. And if this is an interacting amino acid and this is the position of the amino acid, and how about the interacting atoms in the receptor? 1 OE3. These two positions of glutamic acid 257 is interacting with the O6 of verimotry. And the bond lengths for these two interactions are shown here. So this is how we can able to tabulate the results. The next case study 3. I believe I have circulated all these journal papers in the WhatsApp. Okay, go through these journal papers. Of course, I published in open access. You can even download these papers. Even the latest paper, what I work with machine learning. I mean, my and my students. Now let me take you to the case study three. In this case study three, okay, we work with network pharmacology. Okay, so the major things other than the molecular talking, we tried with network pharmacology. Much of system biology we have tried here in these studies. Okay, and if you see here uh, in this study, finally in molecular talking, we have done so many kind of network pathway analysis. So after doing the network pathway analysis, finally we can able to see that in the talking part, out of initially we have taken around 1,000 best receptors for ovarian cancer. Initially, we have taken 1,000 interacting targets for ovarian cancer. Out of 1,000 important 1,000 genes, we have screened the top 10 genes. Whatever you see at the bottom, this is gene 1, gene 2, gene 3. So, like this, we have screened around how many? 5, 6, 7, 8, 9, 10. So, out of these 10 genes, whatever most important for developing the ovarian cancer, we tried talking with a few plant compounds, apigenin, quercetin, resveratrol. When we try to talk, okay, the final results, we can able to find that this quercetin is going to is talk with 6 targets. I mean, the quercetin is giving negative maximum binding energy when compared to the other two control drugs. Then we finalize quercetin is the best drug for the treatment of ovarian cancer. See this. So if you see the best targets, this is one, this is two. I mean, the quercetin which has given the negative maximum. The red color is quercetin. So one, two, three, four, five, six. In all these 6 targets, you can see the quercetin is showing the negative maximum. So we have finalized this is the best. Do you know where exactly we can find plenty of quercetin? So I'm just interacting with you. I shown you a few plant compounds, and you know that our company is selling these plant compounds. We are selling all these 3 compounds. You know what is a source where exactly we can get quercetin? Of course, in food we can find all these plant compounds.
Onions.
What is that?
Onions.
Onions. Very good. The onions you can see plenty of quercetin, and like onions you can see tomatoes. Tomatoes are rich in quercetin. Okay, so whatever we take in sambar is going to combat the ovarian cancer. Do you believe that? Okay, the next thing, resveratrol. Next to that, you can see the resveratrol is the best one. You know what is the source of resveratrol? Where exactly you can find resveratrol? The resveratrol we find in berries. Berries, BRRIAS. Okay, so generally in grapes. Okay, grapes are coming under berries. You can find resveratrol. You see that in grapes, especially the black grapes, you can find the seeds, isn't it? The black grape seeds are excellent anti-cancer drugs. If you procure these drugs are very expensive anti-cancer drugs, and especially they are mentioned like grape seed content is available because that is most effective to treat many types of cancer. And there you can see this resveratrol. Okay, now let me take you to the new chapter called molecular dynamics simulation. So I can't able to illustrate you the MD simulation, but I can tell you how to do it because I don't have a workstation. I'm just illustrating it with the laptop. Okay, but if you want to practice the MD simulation, and if you want to learn the MD simulation, I can suggest you a few colleges. They can train you. Okay, but they charge plenty of fees, maybe 15,000 or more than that. Okay, for 1 month. Okay, and for staying charges you have to pay, like hostel charges, etc. So much demands out there. Even to work with the MD simulation, it looks like a PhD research. It's not like a UGV student, just like that complete using however you do the molecular docking using Autodoc. It's not you can complete as soon as possible. Okay, you have to struggle until you get this. Okay, you have to troubleshoot. You have to clear the box. Okay, troubleshoot with the box, whatever you have. Now let me take you further on molecular dynamics simulation, because this is important. How to infer the results. Even if you get the results from someone, you should know how to interpret the results. Now let me teach you what exactly you can understand from this. Atomic trajectory at microscopic level. Okay, that is actually, you know, the type of mechanics. You remember, I have mentioned you the type of mechanics. Okay, if you want to determine the properties, we have to analyze up to the atomic level. Okay, so for determining up to the atomic level, we go up to the quantum mechanics. Of course, quantum mechanics is defaultly deployed here in this MD simulation software. If you are working up to the microscopic level, it means that we are up to the using up to the quantum mechanics. And if you are going up to the macroscopic level, we are from the classical mechanics. Now, let me take you to the, further, if you see this, I told you Newton's law of motion, F equals ma. Okay. But if you want to calculate for entire, this is for an atom. This is for an atom. But if you want to calculate total force for a protein, okay, that is macroscopic level. It's a protein. In a protein, you have plenty of amino acid. In each and every amino acid, you have plenty of atoms. So whatever you calculate the forces for each and every atom, you sum up all the total forces so that you get for the entire protein. So that's how you can able to see. Okay, for finally, if you actually this is what exactly quantum mechanics. Initially, the fine thing or the atomic level, whatever you study, is the classical mechanics, because we study up to the atom wise. And if you take all these atoms and we work through statistics, then it is statistical mechanics. So finally, in the quantum mechanics, like if you take UV spectrophotometer, if you pass a light, obviously the absorbance is measured. If the absorbance is measured, then obviously from that weight of a protein is determined. So ultimately, if you want to calculate the macroscopic level, we have to start with the microscopic level. Now let me take you to the MD simulation. In MD simulation, we have plenty of softwares, okay, to work with it. But I suggest you to, yeah, if you want to practice with the freeware, try to work with the Chromax. The expansion of Chromax is Groningen machine for chemical simulations. And the Desmond is a AMD simulation software present in Scroid Engine. The recent publication, what we made in scientific reports. So if you see one paper I've circulated like scientific reports 2026. Okay, so that paper you can see MD simulation, work with Desmond, and also no. That also I worked only with Chromax. Okay, but one more paper is going to be published soon. So if the paper is published with Scroidinger Desmond, I'll be sharing that paper also. So in all the papers, I believe I tried with the Chromax. Okay, but Scroidinger has a Desmond uh, tool for simulation. And we also have simulation tools like Chromos. The most widely used simulation software next to Chromax is NAMD, that is Nanoscale Molecular Dynamics. Okay, and yeah, sir, these are other MD simulation tools. If you want to go to the Chromax, okay, simply go with chromax.org. So that URL will take you to this kind of homepage. If you want to download, you can download this. In my ANRF funded project, in collaboration with IIT Madras, we have secured fund to procure a workstation, you know, in protein bioinformatics lab. So in my fund, we procured a kind of workstation, and I downloaded this Chromax and I worked in my funded project. Okay, so we need a funding, of course. Okay, around 3 to 4 lakhs to work with these kind of simulation tools. So in the Chromax, Chromax is a package actually. We have plenty of programs to work in the Chromax. And finally, if you want to see the results, so we have to install an additional tool called XMGR. So can you able to see here XMGR somewhere? Yeah, no, not here. Yeah, X and G are trace graphs. This is the final two, which is connected with, we have to work along with the Promax to find the outputs. Then what are the final outputs you can find in MD simulation? We'll be discussing soon. So before seeing the MD simulation results, what are the different MD simulation softwares or protocols present in Chromax? We have plenty of exe files. Now presently I've shown you the top 12 exe files. The first exe file is the GMX setup. This produces a topology using Chromax for a given structure. The next exe file is the GMX. The next tool is the GMX Solvate. This Solvate is system, and the third is for energy minimization. The fourth is for MD simulation. For positioning strains, we can use GMX systems. Like this, we have plenty of tools present in Chromax. For working with for working with Chromax, we must install it in Linux. If not, if you are working with Windows, again you have to use a Linux interface, and only through the Linux commands you are working with Windows. Okay, you can't use this single click, double click with the mouse. Only thing is that you have to work with commands to work on empty simulation. So if you see the results, okay, empty simulation analysis can be done initially with RMSD. So RMSD is root mean square deviation. So in this example, okay, whatever I've shown, this is actually I've taken from a published journal paper. That journal paper is related to vaccine design. According to this vaccine design, actually I tried talking with a peptide. Okay, this is actually a peptide docked with a protein molecule. When I docked a peptide with a protein molecule, I got these kind of RMSD. So how to infer this kind of a result? The MD simulation I tried working for 200 nanoseconds. In this 200 nanoseconds, so okay, if you draw a line, so if you see this line is stable. If you're drawing a horizontal line, so if you see from here, so around 83 second to 140 seconds, 83 second to 140 seconds, you can find the RMSD is stable. So this is how you can infer the result. And one more thing, if you see this, it comes around 0.55. The RMSD value is 0.55, and you can able to see that it is stable in the time from 83 to 140. Okay, this is how we infer the result of RMSD. Okay, so if you want to know about RMSD, okay, the MD simulation. In MD simulation, I can show you some image. Let me take you to the image. See this molecular dynamic simulation. The a ligand is interacting with the receptor, but it's not in one particular position. Actually, we are running up to some 200 to 500 nanoseconds in a particular time. Okay, so see, again I'm running this. So this ligand is in this conformer, but after moving to few nanoseconds, okay, the ligand has moved. Okay, some kind of a conformation change you can observe. So the ligand is undergoing a flexible motion. Even the protein is undergoing a flexible motion. This part is rotating in actual rotation, and after particular, but you see this, the ligand is not coming out. The ligand is still present in the protein receptor after running to a particular time. For example, if I run across for 200 nanoseconds, the ligand is not coming outside. If the ligand is coming outside of the protein, then you can find the RMSD is unstable. So if a protein, if a ligand is coming outside, okay, you can find the zigzag kind of a motion. So it is stable over this particular time, and here you can't able to find a particular line. So it is unstable. Even if it is stable for a few nanoseconds. Here also you can see the stable thing, but for few nanoseconds. Later you can find the stable for this time. Okay, the line should be straight, it should not be inclined. The RMSD value you can determine from this y-axis labels. The RMSD, if you want to work with the RMSD, the command what we use is the Chromax. Actually, this is the command exactly we have to type gmx spacebar RMS, and then we give the parameter and then give this is an intermediate file. Like however we create map files, TLG files in docking. Similarly, in simulation also we can get the intermediate files. We have to give the intermediate file and the protein file, and finally, whatever we give this rmsd.xvg is the final output file. In this output file, if you want to open and if you want to see this graph, you have to install an additional tool called XM Grace. Do you remember XM Grace? This is what I told you. If you want to see this graph, you have to install an additional tool, and then in the Linux we can open this and we can take this snapshot. The root mean square deviation is nothing but a measure of the average distance between the atoms of superimposed proteins. Now let me take you to the next output. This is what I shared you earlier in the journal paper. Coherent B docking with EGFR and caspase 3. So in the journal paper, you can see this MD simulation. So what is the difference between the previous image and this image? In this image, we can able to see a sample. The sample is in red color. That is coherent B docking with EGFR. So ligand docked with the receptor, okay, is in the bottom, is shown in the red color. The one which is at the top in black color is just the protein receptor alone, EGFR alone. You can able to see the coherent B docked with EGFR complex is showing a less RMST value. Is showing a less RMST value when compared to this EGFR alone. So you can tell the binding of BB with EGFR is stable. The final conclusion you can give is stable. Then, after providing that the stability is found in the binding, then you have to tell how much stable. So you draw a line, you can check. So, actually, this kind of a simulation we work with picoseconds. Okay, of course, this is a 200 nanosecond, I believe. But only thing is that we are showing in numbers like that. Okay, 200 nanosecond. This is 1E405. It means that 100 nanoseconds. We tried only with 100 nanoseconds. And if you see nearly some 20 nanosecond to 45 nanosecond, it is stable. The RMST value is zero point 0.3. One thing, when it is unstable, if the RMST is exceeding 1, if you find RMST somewhere after 1, then the binding is unstable. Okay, this is how we can conclude. And if you see here, the Byron B docked with EGFR is showing somewhat stable result, somewhat stable. But here you can see it's more unstable. So based on simulation studies, Byron B is showing a stable binding with EGFR when compared to caspase 3 receptor. Like this also we can compare and infer the results. Okay, so the next thing is MD simulation using root mean square fluctuation. For this is the formula, and this is an output from a peptide docked with a protein. So RMSF is in angstrom is shown in y-axis. A number of residues. It's not the time in nanoseconds, but the number of residues of the protein is shown in x-axis. So according to this, the RMSF, well, whatever peaks you find at the top is unstable. But if RMSF is exceeding 1, definitely it is unstable. But you see, whatever RMSF peak we find, this is the top peak, this is not exceeding 1. So it means that even root mean square fluctuation from fluctuation graph, we can still tell the that we can find the stable binding of peptide with protein. Okay, so from GROMAX, the results are interpreted like this based on RMSD values and RMSF values. And for executing this, of course, we have to use this command in terminal in the next step. And final output, what we save is rmsf.xvg. So this is another output for the Boirnon B. Talk with EGFR and Caspase 3 for RMSF. RMSF, I told you, we have measure in angstrom in the y-axis, whereas the residues are in x-axis. So if you compare this RMSF of EGFR and Caspase 3, the top peak what I got here is around zero point 0.5. But here the RMSF, we are exceeding, we are exceeding even 1. It means that the Caspase are the BB talk with Caspase are okay. That's what shown in the red color. So just because it's crossing 1, it is unstable. This is unstable. The binding of pyrimidine B. So it means that pyrimidine B is coming outside of the caspase 3 after a particular time. Okay. But whereas here it is stable because the RMSF is less. Okay. Fluctuation is very less. Now let me take you to the next analysis is radius of gyration. The radius of gyration is represented by Rg, and the radius of gyration, if you want to work with it, this is the command we use. So radius of gyration, you have to check whether when it is stable. So you can see the radius of gyration nanometers shown in y-axis, time in nanoseconds in x-axis. So this up to this particular time to this time, it is stable, and similarly this time to this time, it is stable. So that alone I can mention. Okay. So this is how we interpret the results of radius of gyration, which determines the moment of inertia. You remember, just now I showed you a video that a ligand is talking with a receptor. When it is doing the 200 nanoseconds, the ligand is moving, and even one part of the ligand, the cyclic ring, is also rotating along the axis of rotation. So, whatever we do in the MD simulation for this, which is rotating axis of rotation, that's what radius of gyration. Remember how it is rotating? The final time this is rotating in the axis of rotation. So in that case, we can able to measure through radius of gyration. And the next important interpretation can be done through finding the binding energy. Not only we can find the binding free energy from Autodoc, but we can also make it using MD simulation. In binding free energy determination, we can able to determine through three approaches. One is linear interaction energy approach, which utilizes two trajectories approach, and we can also see MMPBSA, which is using one trajectory, MMGBSA with three trajectories. What is that one, two, three trajectories? See here, whatever we see here, in the yellow color is the receptor, and whatever we see here is the ligand. In the one trajectory, we need just the energy of ligand and receptor complex. This is for MMPBSA. For linear interaction energy, we need two molecules. One is ligand receptor interaction energy and the ligand alone. So these two together we measure through two trajectory energies, and that's what we calculate in linear interaction energy. The third thing is the three trajectories, that is receptor, ligand, and the receptor alone, the ligand alone. This is what three trajectories. So during the three trajectories, if you want to calculate the binding energy, it is calculated as a del G complex minus del G receptor minus del G ligand. So this is applicable for three trajectories. So you can find the expansions. What is MMGBSA? First, we have seen this one trajectory as MMPBSA, isn't it? That is molecular mechanics Poisson-Boltzmann surface area. And the next thing is GBSA, three trajectories is molecular mechanics generalized bond surface area. LI is linear interaction. MD simulation, actually, we are talked with multiple compounds. We get like this kind of a graphs. Okay, so we are trying to publish this kind of results. Okay, I have completed MD simulation, but still, how to perform, how to after installation, how to perform MD simulation. Okay, so for this, we can go through these tutorials. Give me just 1 or 2 minutes to complete this MD tutorials. Okay, I have shared you this link. If you click that link, it will directly take you like how to practice the ligand receptor simulation through the tutorial. So after procuring the workstation in IIT Madras and after installation of Promax, only this tutorial helped me to how to practice with this protocol. Okay, entire protocol is given. So you can see the steps 1, 2, 3, 4, 5, 6, 7. Finally, the seventh step is analysis. Okay, the detailed procedure is mentioned here. So see here from the first step onwards, you are using the commands, Linux commands. Grep is the filter command. Okay, and tar is a command used to zip the file. You heard about WinZip file, WinRAR file. These are compressed files, isn't it? Similarly, if you want to compress the files in Linux, we use a command called tar. Okay, so this is how we can compress the files. So there are a lot of, you should first understand how to use the Linux commands. And you should also have prior knowledge on Perl programming and Python programming for this. So these skills are also needed. So when I'm telling you, working with MD simulation is this kind of this research is quite equivalent to PhD research. Okay, so how to use the GMX command? I was explaining, if you want to convert PDB to GMX file name, okay, that is Promax file name, then you have to use this command accordingly. Okay, so this is the step you make, and after making this step, you can find this. So the next step is ligand topology. You have to simply click that hyperlink, then it takes you to the next thing. So here it asks you to download the Avogadro program, and earlier it asked you to use the Python program. If Python program is not compatible, you have to change the Python programming code there, and then you have if you work, then it works. Okay, this is the detailed protocol. One can work with AMD simulation using Chromex. So after giving an input, how you get an output, and after giving a next input, for how you get an output. So like this, each and everything, input and output details are mentioned in these pages. How to build the topology. Okay, and this is like solvation. After building the topology, the next step is to define box. That is, you are defining a box for the protein molecule, creating a grid box, and then you have to solve it. The purpose of solving is to determine the binding sites and even how well the conformation of ligand binding with the receptor is going to change. So all these things we can able to make it. So these are the commands we can use. Okay, and then adding ions. Even in Autodoc, we add charges, we add hydrogen atoms. Similarly, here also we can do like this. Okay, so and then energy minimization protocol. In docking, you have done. Even in MD simulation, you can do this. Okay, and then equilibration protocol, and then you can go with the equilibration like under a standard number of atoms. What is the standard temperature and pressure we keep for simulation? Okay, we use n stands for the number of atoms, v stands for volume, t stands for temperature. We go with nvt equilibration and npt equilibration. So n stands for number of atoms, p stands for pressure. So here it is volume, p for pressure, and t for temperature. So that is the second thing, NPT equilibration, after NVT equilibration. So after doing all these simulations, equilibrations, then we go with the production MT. So this is the command we use. If you use this command, and finally we go with the analysis. The final analysis, so you can see a ligand is binding with the receptor. And after talking this, if you analyze the results in the form of RMSD, RMSF, like however I showed you the results, you get an output like that. Got it? So I have completed this session on MD simulation. Like if you work with work with simulation, I told you to install the Promax in a workstation, and to work with the initialize sample file, work with the tutorial file present in the URL pasted to you. Any doubts you can ask me. Do you have any doubts?
Sir,
Yeah, ask me.
Romax can we use it on our laptops, sir?
It's not possible. I told you. So we need a high configuration storage space and processing power, including we should have a graphics card. Okay, high capacity graphics card.
Okay, sir.
If no other doubts, thank you very much for your patience and interaction. Would
Hello.
like yes, madam. Yes, madam.
Sir, may I know which are the institute or colleges offering this MD simulation internship or for
Um,
performing this kind of
no,
activities and giving the result?
madam, actually, you can request through personal WhatsApp. I can provide you this kind. Okay. So, whoever requires, you can request additional services from other institutions through personal WhatsApp. I can direct you about it.
Sure, sir. Thank you, sir.
Thank you. So, any other doubts from any other participants? If no doubts, thank you. Okay. See you then. Bye.
Thank you, sir.